2 섹션 진행하도록 하겠습니다. New Technology to Improve Laboratory Productivity입니다. 많은 도움 되시기 바랍니다.
I'm going to start the 7th afternoon session with new technologies to improve laboratory productivity. So in this seminar, I'm going to talk about the bottlenecks and other sources of inefficiency in analysis workflow, the strategies that to eliminate or reduce the impact of those inefficiency, and the cost and the benefit of adopting those strategies. A typical laboratory workflow consists major of 4 steps. That's sample collection, sample preparation, analysis, data management, and the reporting. The time breakdown of the 4 steps may surprise you because the majority of the time is now spent on the analysis. You can see only 6% of the time, of the total time. Instead, a majority of the time is spent on the sample preparation and data handling and the reporting that cost 60% and 27% of the total time. So we can identify the bottlenecks and the target area for improvement, such as throughput, cost control in solvent, consumables, and maintenance. We also can target for better results in terms of accuracy and reproducibility. The improvement in this area can be linked to technologies and support from a partner like Thermo Fisher Scientific, because we are devoted to improving food safety. The challenge in the process of improvement is you get reliable results at reasonable cost. So the first bottleneck in the laboratory analysis workflow is sample preparation. Does your laboratory prepare sample very efficiently? How can you prepare sample efficiently? And what's the technology and the solution Thermo Fisher offer you to reduce the impact of this bottleneck? Let's review the modified catcher method I talked in the morning. The modified catcher method was invented in 2003 by a USDA scientist, and now it has been widely applied for analysis of maltol residue pesticide in fruits and vegetables which contain high moisture content. This method is very efficient, very simple to use. In the workflow, we modified only 15 steps. For 12 to 40 pre-weigh sample, only takes 2.5 hours to prepare the sample. The consumable you use using this sample preparation, only plastic tube, salts, and solvents. You don't use very expensive laboratory equipment for preparing the sample using this catcher method. So besides the catcher method, what's the other solution that Thermo Fisher Scientific offered for pesticide analysis? For volatile pesticides, we recommend you use SPME. SPME represents solid phase microextraction. It's a very simple sample preparation technique. For volatile pesticide analysis, we recommend you use headspace SPME. When you use headspace SPME, you just expose the SPME fiber, let's fuse the silicon fiber to the headspace above a sample. The volatile pesticide evaporated to the headspace of the sample will be selectively absorbed onto the fiber, which coated with polymers. So after the extraction, also concentration of the volatile pesticide on the SPME fiber, you take it out, then inject to a GC system, then thermally dissolve the volatile pesticide on the fiber. For our application, the manual sample preparation part takes less than 2 minutes. The 2 minutes required for weighing the sample and place into the sample vial, adding some water. Because from after the sample is weighed, the SPME outer sampler will take over. Extraction, concentration, injection, all automatic. For our application, we use 30 minutes incubation time using a head-based SPME. No solvent is needed. We use the head-based SPME method to prepare for analysis of volatile pesticide in tea. We include 43 pesticides belonging to 16 different chemical classes. We choose tea because, as I said in the morning, tea is a very complicated, challenging matrix. If you use other sample preparation method, may require several SPE cleanup, which is very labor-intensive, time-consuming. However, using our head-based SPME method to prepare the sample for volatile pesticide analysis, the manual sample preparation part only takes 2 minutes. Other part automatic. And we also coupled the head-based SPME with our TSQ quantum GC. That's the very sensitive quantitative detector. You remember we coupled our modified catcher method with ion trap MS detector. However, if we couple with the head-based SPME with ion trap, the ion trap is not sufficient for the head-based SPME to extract enough volatile pesticide to be detected. That's why we couple the SPME with the more sensitive TSQ quantum GC to get higher sensitivity. Because the higher sensitivity and the higher selectivity of TSQ quantum GC can offer, using very simple head-based SPME method, all the 43 pesticides get LOD lower than the 10 ppb. That's the EU MRLs for pesticide. Here I show 4 calibration curves for 4 pesticides. Using the head-based SPME technique to prepare the sample, we get excellent linearity over several orders of magnitude. Now I'm comparing the head-based SPME technique with manual sample preparation. The SPME method, you can do extraction and concentration in one step. So it's very simple sample preparation technique. When you use head-based mode, you don't need any solvent. It's solventless. Also, when you use SPME, you can do head-based mode, also liquid extraction. That means you dip your SPME fiber into the liquid sample to extract the compound you are interested in. When you use head-based SPME mode to prepare volatile pesticides, because the matrix interferon compound, most of them not absorb onto the SPME fiber, so there's less matrix interference to your GC system, to the injector column, also your MS system, so less maintenance required for the MS source and the GC column. This head-based SPME sample preparation is working well for volatile pesticide in low-fat matrix, such as fruits, vegetables, coffee, tea, and non-dairy beverages, especially when you couple it with our sensitive TSQ quantum GC, you get very high selectivity, also sensitivity. The cost associated with adopting the head-based SPME method is you have to buy a SPME auto sampler that will give you much higher reproducibility for sample preparation. And you need to buy a SPME fiber. Each fiber you can use minimum 200 times. And the benefit you get is this technique is so simple, so easy to use. As I said, the manual sample preparation part only takes 2 minutes. The later step will be operated by the SPME autosampler, so you get very high sample throughput. You can reduce matrix effect because you use a headspace SPME, and the matrix compound will not be absorbed onto the SPME fiber. So those benefits will save you money in time, labor, and the solvent, especially when you use acetonitrile as the extraction solvent for pesticide. I think if you work in the lab, you know acetonitrile was so expensive last year. One bottle can cost several $100s. So you can get very good benefit using this solventless headspace sample preparation technique. So in next several slides, I'm going to show you different chemicals, veterinary drug and pesticides belong to different chemical classes. So here belong to 7 chemical classes, total 49. Actually, we should show total 54 pesticide and veterinary drug. So the first chemical class is macrolides. I show one example here. And phenycols, one example is chlorophenycol, beta-lactam, and aminoglycosides, acaricides, sulfonamides, tetracyclines. So from the different structure, you see those compounds have very different chemical natures. So can we use one method, one sample preparation method to analyze all these compounds and in a very high throughput way, automatic way? Yeah, if you have such method, the method would be very overwhelming, right? But we, Thermo Fisher Scientific, has one technology can analyze all the compounds which belong to different chemical classes. This technology we call turbo flow technology, or turbulent flow chromatography, or TLX. So they mean the same thing. Turbo flow technology, turbulent flow chromatography, or TLX, same technology. So now let's compare how efficient turbo flow technology for screening those compounds, comparing with manual sample preparation. Here is the workflow using manual sample preparation for quantify 5 compounds in honey and analyze those compounds by LC-MS for 12 samples. Takes 7 hours. Using manual sample preparation technique for 55 compounds in honey. This is the workflow for manual sample preparation analyzed by LC-MS 12 samples. Takes 5 hours. However, using our automatic sample preparation technique called turbo flow technology, this is our sample preparation flow rate, and TRX represents turbo flow technology, analyzed by LC-MS 12 samples, only takes half hours. So you can see your sample throughput improved 10 times compared to this manual sample preparation. How turbo flow technology can do that so efficiently? The mechanism is, the magic is turbo flow technology using a turbo flow column to clean up your sample. The turbo flow column is based on 2 separation mechanisms. One is traditional column chemistry, like what you have in an LC column. If you have a reverse LC column or normal phase or ion exchange, etc. So the turbo flow column also have the same type chemistry on the column. In addition, turbo flow column has a size selectivity, that's size exclusion mechanism on the same column. The size exclusion mechanism is based on that when you use turbo flow technology, you run the flow rate very high. We run generally 2 ml per minute. And under such high flow rate, small molecules diffuse faster. They can diffuse to the pores of the turbo flow column particles. So stay in the column longer and separated. And the large molecules, that's the matrix interference, does not diffuse slower, does not have enough to diffuse to the pores of the column particle, so will be swiped out to out of the turbo flow column. So only the small molecules, that's the veterinary drug and the pesticide, stay in the column separated. So that's the size exclusion mechanism to separate matrix interference and your target compound using turbo flow technology. So turbo flow column has 2 mechanisms on one column. That's unique, different from the traditional LC column, only one mechanism in one column. So next, I'm showing you another example using our turbo flow technology to clean up and analyze veterinary drug. We choose the analyzing the veterinary drug leucomalachite green in shrimp. Malachite green is a toxic dye. When it's diluted, it can be used to treat disease in seafood. However, malachite green is carcinogenic. It's banned for use in aquaculture. But after malachite green is absorbed by shrimp or fish, it's converted to the colorless form, leucomalachite green. So we use the turbo flow technology to monitor the leucomalachite green in shrimp. Shrimp is a very complex matrix too. However, using our turbo flow technology, no problem to clean up the matrix interference. You can see, compare this chromatogram is the sample extract for leucomalachite green without any cleanup, and this one is the sample extract of leucomalachite green. After turbo flow technology cleanup, the signal noise increased is 331 compared to this signal to noise ratio 59. So after cleanup, turbo flow increased the signal to noise ratio dramatically. So remove your matrix interference and the background noise. So the cost of adopting turbo flow technology is you have to buy